So today I was asked to present the work that I've done in the two European projects. The first one is Seamark, which will end at the end of June, and the second one is within the Interreg project Marine Aquaculture. So we work for, if you can go to the next slide, please, Melanie. So just to tell a bit about the context, Seamark project is a very big European project that will focus on the several supply chain for algae with two species, Saccharina Latissima and ulva. And the project aims at improving each step of the supply chains to go from material, so biomass, to end product. So as the entry of the supply chain, the biomass and the selective breeding to improve the biomass was important, and that is the part where the Station Biologique de Roscoff was involved in, along with partners for ulva in Portugal, in Ireland, sorry. So we worked with two producers, one in Brittany called Algolesco, and one in the Faroese Island called Ocean Rainforest. So I will talk more about that. So if you can go at the next slides, please, Melanie. So first, our selective breeding program in Seamark is based on genetic resources, as every other breeding program. So we build on the collection isolated during Genialg, and we also widen it with sample from the three partners that were in the project. And we have added 9 sampling sites and we also have cultivated species and cultivated strains, so meaning that there has been derive on the lines and some wild resources. So from these samples, we isolate gametophytes, so male and female separately. And we got a collection of about 300 fully sexed and clonal gametophytes. They were vegetatively propagated, and they will be soon deposited in the Roscoff Culture Collection in Roscoff to be publicly available for other scientific projects. If you can go to the next, please. So just the one before, once we got the resource, we made some controlled crosses. So one male, one female, randomly across the population, to make them reproduce, and you can see on the pictures a small sporophyte growing in small petri dishes at first. Next, please. So we had one major constraint. It was no introduction of alien polymorphism in the site. And in Brittany, we have the obligation of cultivating local strain. So the questions that were raised were can we breed without introducing non-native germplasm? Being known that in plants breeding, the crosses often involve wild strain from all over the world, and this we can't do it with algae. Will be the cross fertile, as fertile when we go from within one locality or between two localities, and do we have heterosis or outbreeding depression? So to answer this question, we made 4 types of crosses. The first one was intra-Faroese Islands, so one male from Faroese, one female from Faroese, the same in Brittany. And we also made some between crosses, so one male from Faroese with one male of Brittany, one female of Brittany with one male in Faroese, so that we can compare between inter-regional cross with intra-regional cross. Next, please. So regarding the cross success, as you can see, there is no significant differences between the intra-region crosses and the inter-regional crosses. We also confirm that we can make fertile gametophytes that were kept in collection and in vegetative growth for 10 years, because genial collection was isolated around 10 years ago. Next. So once we have the sporophyte growing, so it's the first generation, We followed the growth from the small size of plantlet of few centimeters up to almost 1 meter. And we had a last experiment where all the blades were coppized at 30 centimeters above the meristem and let grow in a common garden experiment where we followed them every week. So to measure the growth, we took pictures of the blade every week, and we followed the blade area to calculate the specific growth rate. Next, please. Thank you. So these are the results. I think that there is a slide before, Melanie. Yeah, so for the growth performance, okay, the graphic is not showing. So we got a nice variability in the growth rate. And most importantly, we only have one type of cross that was significantly less performant than the all over three. It was a cross from ocean rainforest, so from the Faroese. But there were no differences between the crosses intra-Brittany and the two other inter-crosses that we made. We hypothesized that the Faroese strains were less adapted to our cultivation condition here in the lab because it was maybe a little bit hotter and the photoperiod might have been quite different as well. Furthermore, we investigate the biomass quality, which is very important to cultivate algae, that they have to be good quality so that its product at the end is you can have it with good yield. So this is a result for the alginate content in the first graph. And the second one is the composition of alginate between M motif and G motif. So you have on the right side the means of all the crosses. So ALO and ORF are the intra-regional crosses, and ALF and ORA are the inter-regional crosses. You can see that we have slight that the intercrosses are slightly better than in terms of content, but also of quality, than the intra-crosses sporophyte. But the difference is not very significant, meaning that the ALO, for example, crosses are not significant different from ALF and ORA as well. So I think it's my last slide. So the conclusion would be that yeah, sorry, no, I'm not done yet. So once we got the performance from both growth potential and biomass quality, we selected the best parents that were sent to the farm for farm cultivation evaluation, yield evaluation under farm conditions. And this is ongoing. And this is it's only after this cultivation that we will know if we got some genetic progress or if the crosses, the F1 that we made will be a bit better than the ones that are already cultivated. Can go to the next, please. So this is again the breeding scheme that we had in Seamark, and we identify bottlenecks. So if you can click, Melanie, please. And bottlenecks that we identify were the preservation of genetic resources for breeding program. We also have a little information about gametophytes phenotype, when we could select phenotype directly on them, and it would be easier in terms of logistics, but also maybe quicker. And the last, not the least, was a very slow growth of isolated gametophyte. So we designed a pilot project called kelp-secure, where we aim at biobanking and cryopreserve the gametophyte, so the genetic resources, phenotype for growth performance of the gametophyte, but also for biomolecule content. We try to optimize growth condition, culture condition for growth of gametophyte, and lastly to upscale gametophyte cultivation using bioreactors. So I won't go into details, so my presentation won't be too long. If you can go to the last slide, please, Melanie. So thanks to this pilot, we had some nice results. So we have protocol to efficiently cryopreserve the germplasm. With the culture optimization, we could detect that gametophytes are very sensitive to high light and high temperature, and they thrive in narrow range of conditions that have to be refined still. And we also end up developing dedicated dedicated system to scale up gametophyte growth with an automatic fragmentation system. I know that there are some such device in America, but this has been developed with French companies. And I think that I've said everything, so thank you for listening, and if you have any question, I can try and answer them.